Okay, let's try this again. Okay, so welcome to Physiology videos. So the goal behind these videos specifically is to complement, kind of go over all the material given by the current professor, as well as just kind of add and clarify some different concepts and some of the material just to make sure that we really understand everything and build a solid foundation within physiology. The really important thing about physiology is that your next couple of trimesters, you're going to start looking into pathologies. And in reality, that is what pathology is really just a study of how normal physiology is messed up or something is going wrong with your typical physiology. So it's really, really important in understanding these concepts and these ideas to be able to later understand disease process and specific diseases. Okay, and I personally really love physiology because physiology is all about kind of a flow chart of the body, where if one specific thing happens, it's going to cause something else to happen, or if this doesn't happen, then something else is going to happen, and it's continuing to go on and on and on and on. All of this material is really going to be kind of in a way cumulative. I don't know exactly if it will be tested cumulatively, but if you have, in order to understand this stuff further on down the road, you need to have this solid base here at the beginning. So what we're going to be talking about first is membrane physiology. Now, full clarity, this is the second time I'm recording this. The first time the audio got corrupted, so I'm having to re-record this. But I'm going to try and include everything that I did in the first recording, and which is kind of nice because then I already have all of my notes added in. Okay, so first we're talking about diffusion and osmosis. These are different things, and I'm actually want to cross off osmosis on this one. We're just going to talk about diffusion first. So diffusion specifically is the movement of solutes from high to low concentration. So solutes specifically is talking about the stuff that's mixed into the liquid. The liquid is called the solvent. You can have proteins or the different ions, cations like sodium, potassium, and calcium, or an anion like chloride, as well as gases like oxygen and carbon dioxide, and et cetera, et cetera. A whole bunch of different things can be solutes, can be mixed into that aqueous solution or the liquid, the solvent solution. It's always important to understand that with diffusion we're going to be going from a high to a low concentration, meaning that just like this little picture shows, if we have a higher amount of stuff in one side, it's going to travel to where there is less stuff mixed in. Okay, and this happens because our membranes, specifically our phospholipid bilayer, hopefully you remember that from biochemistry, where we have the hydrophilic phosphorus heads and the hydrophobic lipid tails, and all of our membranes where they're going to control what goes in and goes out, and what we have inside and outside of all of our cells, kind of where everything is. And that membrane specifically is called semi-permeable, meaning that it keeps certain things out, but other stuff can get through just fine. And the things that aren't able to pass directly through the membrane are going to need help. They're going to need what's called facilitated transport. Facilitated meaning they require a protein, specifically an intramembranous protein, to help them cross. We talked about intramembranous proteins when we were talking about biochemistry, as well as quite a few examples of those intramembranous proteins when we talked about stuff like the different shuttles going through the body, where we needed to get specific intermediates from across certain membranes. And so they did that by kind of transporting across, like giving one thing across and bringing one thing back. So you can have intramembranous proteins that sort of do and play that role in helping to get certain intermediates and certain solutes through membranes. Okay, so talking about membrane transport, we really actually, I'm going to just skip this real quick and talk about osmosis. So with osmosis, diffusion, we talked about the solutes, or the stuff, moving from one place to another, the membrane. Osmosis, we specifically talk about the water, the H2O, that goes across that semipermeable membrane. Okay, so similarly, we can have right here two things of a solution with the same amount of solute in it, but because there's a different amount of solvent, there's a different amount of solvent, different amount of water, it's going to be a different concentration. So in the concentration is, this should be the solute. Yeah, this should be solutes over solvent. Concentration, so the concentration is the solutes over the solvent. So it's how much stuff is mixed into how much liquid. And that gives us our concentration. And we want the concentration on either side to be as close to equal as we possibly can get it. Okay? So typically the water is going to move to the area of higher solute concentration in order to kind of balance it out. And you should have talked about this a little bit in biochemistry as well as in cells and tissues, where we have something called an isotonic solution, meaning that the concentration is the same inside of the cell and outside of the cell. Hypotonic, meaning that there is less inside than outside, or there's more solute. So hypotonic solution means that there is higher concentration of solute, or of stuff, inside the cell. So water goes in and the cell gets bigger. I remember this because hypo sounds like hippo. Hypotonic, hippo gets big, getting bigger. Whereas the hypertonic solution, meaning that the solution itself outside of the cell has a much higher concentration, so it pulls water out of the cell and you get shrinking of the actual cell. Okay, jumping a step back. So that is what we're talking about, osmosis. Osmosis is the water moving. Diffusion is the solutes, or the stuff moving across the membrane. Okay, please remember that, because they're very important to keep those separate in your brain. Okay, so now we start talking about a little bit more about membrane transport, specifically when we talk about such as like diffusion. We have two different types. We have passive transport and we have active transport. Okay. We also have what's called simple versus facilitated. Okay. Simple just means that there are no membrane proteins, or no proteins or channels or anything involved with the diffusion process. So the things go in and out across the membrane without the help of any protein. They just are able to slip right through. Facilitated diffusion I'm gonna have to clarify on this specific one, because it's been my understanding that simple diffusion does not involve any protein whatsoever, and facilitated diffusion, even channel diffusion, is considered facilitated diffusion. Let me real quick. Yeah, I believe that this picture, clarify with your professor what they want you to understand about this. However, other sources will say this, that the channel proteins, gated channel proteins, and carrier proteins are three types of transport proteins that are involved in facilitated diffusion. I've always understood that if you're using a protein, so this is facilitated diffusion, and just this is simple diffusion. That's how I have always understood it, and that is the way that I personally would go about stating that. Okay, continuing. So when we talk about diffusion, we have we're talking about simple and facilitated. Simple being that there is no protein involved. Facilitated being that there is a protein involved. Either a channel protein or a carrier protein is involved with facilitated diffusion. Now, that non-facilitated and facilitated can also be passive. Passive meaning that there's no additional energy required, meaning that there's no ATP used. ATP is our main source of energy in the body. So no ATP being used means that there's no additional energy required. Okay. And again, this is all going to occur because it's passive. It's going to kind of go with the flow, and it's just going to occur down a concentration gradient. When we talk about a concentration gradient, that's just like what we were talking about here, where we have more concentration on one side of the membrane and less on the other. And so you go from the high to the low concentration. So there's different types of passive transport, such as directly through the phospholipid bilayer, that is our non-facilitated, or you can use a protein channel or a carrier, and both of those are our facilitated. The intramembranous proteins, again, these channels or these carrier proteins are proteins that span in between the membrane and help carry things across. Besides passive transport, one more thing actually. So when we talk about passive transport, it can combine with our simple and our facilitated. I believe I wrote that down here. I did not. Let me make this an official note onto here. So for example, with passive transport, let's say we just have simple diffusion where it's just going straight through the membrane. This would be considered, you can call it simple passive diffusion, is solutes going straight through the membrane with no help. That's our simple passive diffusion. Then we could have facilitated passive diffusion. And that is solutes going through an open channel protein. Another example of facilitated passive diffusion, same thing, would be the carrier protein helping a solute cross the membrane with no ATP used. So see, we can see the different examples. We also have what's called active transport. So the difference is that active transport requires ATP. It is ATP dependent. These are always going to be facilitated, and it's going to occur against a concentration gradient, meaning that it's going to go from low concentration to high concentration. And that's why it needs the ATP, because it requires the energy from the ATP to kind of push against that gradient. I think you can think of it like it's trying to swim upstream or go upstream, so it requires energy to go upstream and to go against the current of like the river pushing it back against it. Another example, this example that I drawn was talking about how passive versus active, where with diffusion, it's like the rock on the top of the hill. Once it steps off, it just happens. So it can go from high concentration to low concentration without anything helping it. Whereas active transport, you need to actively put energy into it to go from a low concentration up to a high concentration. We already talked about osmosis. This is another slide going back talking about simple versus facilitated diffusion. We already talked about this before, but just to clarify again, simple diffusion is where no intramembranous proteins are being used. And facilitated diffusion, some sort of exchange protein or channel is being used in order to transport ions or compounds or anything across. So active transport, this is going to be the most important active transport protein, intramembranous protein, that you're going to see. It is the sodium-potassium pump, also known as the sodium-potassium ATPase, or you can see it written as the Na plus K plus pump / ATPase. Any kind of combination of those is going to be specifically this protein, this specific, what is happening with this specific example of membrane transport. It being active transport, we know that active transport means that it requires ATP. That's the big thing right down here. It's using energy to push against concentration gradient. And specifically that concentration gradient, there's two of them. It's the sodium and potassium concentration gradient. So this is an example here. It said a banana in the ocean. This is how I remember the normal concentrations of sodium and potassium. So if you imagine that the banana is the cell, but the bananas are full of potassium. So inside of the cell, you should have high potassium concentration. Okay? Inside of the cell is high potassium concentration. If you put the banana in the ocean, oceans are salty and it has high sodium. So there's a high sodium concentration outside of the cell. So outside of the cell, it's sodium. Inside is potassium. And that's there's going to be changes in that due to exchange with ion channels that we'll talk about later on with some of like the interactions and some of the reactions that we have that occur. But that's why we have this sodium potassium pump. It's its job to kind of bail out the sodium and bring back in the potassium as we have different channels and stuff exchanging these different ions to have different reactions happen. And so inside of the cell, we want more of this potassium. Outside of the cell, we want the sodium. And so there's an exchange that with each ATP, we pump out 3 sodium from the inside to the outside, and we bring in 2 potassium from the outside into the inside. This is typically something that gets tested fairly, at least 2 or 3 questions always end up getting asked specifically about the sodium potassium pump, just because it's really crucial in its role of maintaining membrane, resting membrane potential. Also written as RMP, resting membrane potential. And we'll get more into what that exactly means further along in the presentation. I'm just going to introduce it right here. Okay, so when do we have a couple of different channels, and we saw up here how we have our proteins and we have our channels. So now we're going to jump into looking at a couple of these different channels. And these channels specifically, these leak channels and these gated channels, are going to be passive channels, meaning that they're going to go from a high concentration to a low concentration. They're going to follow the concentration gradient. So we have leak channels. Leak channels are channels that are going to be constantly leaking. They're leaky channels. They're going to constantly be leaking ions or solutes across them typically at a slower rate. And that's determined by their size, shape, and the charge of the channel and the ion. So there's all these things, kind of like how we talked about in biochemistry, how enzymes are really specific to each other. A lot of these channels are also going to be specific to have that specificity to specific ions or compounds or molecules or that sort of stuff. So based on the shape of the channel and the shape and also like the charge and the polarity and everything of the channel itself is going to determine what sort of ions or compounds or whatever is allowed to go through it. So it's determined by the size, the shape, and the charge of the channel and the ion. This is also going to be passive facilitated diffusion because it's going from that high concentration to low concentration using a protein channel. We also have our gated channels. We have voltage gated channels, mechanoreceptor channels, and chemically gated channels. These different 3, we'll talk about them in different times. Big ones that we're going to focus on are voltage and chemical gated channels. So our voltage gated channels, we're going to be seeing and talking about those a lot when we talk about action potentials, which is how our nerves communicate with each other and pass messages along. We'll get into that later on in this presentation. And also mechanoreceptors, which open response to deformation of the membrane. So stuff like, even like touch, like this can cause mechanoreceptors and stuff to go off, or certain other movements within the body can cause mechanoreceptors to go off. And that actual physical movement causes those channels to react, to respond, to open, allowing the exchange of different ions and different compounds and stuff to allow the body to sense and to send messages, and for the get those messages into the brain and where else they need to go to allow the body to respond. So really cool stuff. And then finally, chemical. The chemical channels. We have something called ligands. We talked about this a little bit in biochemistry. Specifically, a ligand. Let me pull up a good definition. So a ligand is an ion or molecule, specifically this, a molecule that binds to another, usually larger molecule. So it's going to be some sort of molecule, a chemical, that's going to go and bind onto a receptor, onto another molecule. In this case, a lot of what we'll talk about is that the ligands, such as acetylcholine, norepinephrine, epinephrine, GABA, or glycine, are going to go and bind onto an intramembranous protein and cause that intramembranous protein, especially like a channel, to open and allow the passage of different ions and different things. And that is our chemically gated channels. Okay. So this is kind of our intro to membrane physiology. I'm going to go ahead and stop this recording here. I want to keep these a little bit shorter, just so that way you can kind of pick out specifically what you need and hopefully not get too overwhelmed with these. Again, big concepts to remember. What is the difference between diffusion and osmosis? Diffusion is the movement of solutes. Osmosis is the movement of water. Passive transport versus active transport. What makes it passive? What makes it active? Simple diffusion or simple diffusion, or facilitated versus facilitated diffusion. Which of those, what does that mean? Simple diffusion means that there's no protein being used, no intramembranous proteins were used, versus the facilitated, that it's some sort of exchange protein or channel is being used in order for that exchange to occur. The sodium-potassium pump, please remember this. And then, of course, the different types of gated channels. We will continue to get into these. This is just the introduction for it. We'll get deeper into those when we talk about now our membrane potential and everything that's going on in the next video. Thank you so much for watching, and have a great day.